All right, my name is Tim Strathman, and I'm on the faculty here in the Department of Civil and Environmental Engineering, and also a member of the Center for Advanced Materials for the Purification of Water with Systems, which is a National Science Foundation Science and Technology Center that is centered here at the university. As an environmental engineer, or I should say earlier today, we've had a lot of good talks on occurrence, biological effects, also some studies on talking about some techniques that can be used to look at fundamental surface interactions, which are going to be quite important to understand some of the processes that I'm going to talk about. But as an engineer, we also want to start to think about the fate of pharmaceuticals during conventional drinking water treatment processes. And also maybe in certain situations we might want to think about the next generation of technologies that might, in what I would say, be able to possibly target those micropollutants that maybe the toxicologists tell us are of greatest concern, because I don't think we're going to end up just saying we're going to blanketly treat for everything until it's below detection limit. I'm going to avoid giving any introduction. I think we've had a good amount of that today. Everyone knows that I think agrees that pharmaceuticals are an issue. But I do want to talk about some of my group's research questions and where we come into play in this. And I want to talk about three particular projects that we've been doing. One where I would say it's maybe looking at chemistry that occurs in a conventional treatment process that is used today. And then two projects where in our center where we're looking at maybe the future, our next generation of technologies that we want to try to develop as we go to the future. And there are certainly a lot of challenges to making those commercially viable, and we can talk about some of those as well. And then I want to just finish with my thoughts on some of the challenges to treating water containing pharmaceuticals. So where does my group come into this? Well, we are interested in identifying improved strategies for treating pharmaceuticals and also other wastewater-derived micropollutants that are ending up in our surface waters because they're incompletely treated using conventional treatment process. I personally have a background and specialty in redox transformation processes, so my group is focused on redox mechanisms for transforming contaminants. Certainly, there are other physical separation techniques, membrane treatment processes, things that I'm not going to talk about that we don't do that, but there are certainly other groups looking at those things. I'm particularly interested in processes that can be made to be compound selective. That is, if you tell me that endocrine disrupting hormones are the real bad players out of all these pharmaceuticals, maybe we can, if we know something about how we can treat those chemicals that other processes for which those chemicals are going to be sensitive but other chemicals won't be, maybe we can target that contaminant that's present at microgram or nanogram per liter levels in water that contains milligram per liter levels of organic matter as a general rule. And also anything that we develop, we want to think about things being sustainable. And to me, sustainable can mean that if it's a process, it's something that we isn't going to die off. If I'm talking about surface mechanisms or materials-based technologies, surfaces can't be fouled and lose their activity after one day or 1 week. And also just the energy inputs. We're going to have to think about any technologies that we want to address. If you want to remove nanogram per liter pharmaceuticals and you realize, well, we can do that with existing technologies, but the energy input we have to put into that today maybe we have to think about this. Or there's also a lot of questions these days about contributions of greenhouse gases from energies. So anytime we do something, we probably should think about that. We've been looking at some just fundamental kinetic studies and transformation products to try to understand some of these different transformation processes. And also we have questions about if we're treating real waters contain, again, a lot higher levels of other things that we just naturally occurring organic matter. And we want to know how those things and other constituents in water are going to influence process efficiency. So the first project I want to talk about is looking at the oxidation of pharmaceuticals with permanganate. This is a project funded by the American Water Works Association Research Foundation. And our goal in this project generally is to try to assess pharmaceutical fate during conventional treatment processes that we're already using, other groups have been looking at things like chlorine and ozone processes that are used, oxidizing agents. But very little was known about what might happen during permanganate treatment processes that are used at a number of plants for manganese control, for taste and odor control problems, So probably the majority of plants don't use permanganate, but there is a sizable fraction that do. And so we wanted to know what's going to happen. What's going to be the influence of these processes on? So we're not necessarily looking at it as a treatment process that we would engineer for this reason, but I guess that could be used if we wanted to in certain cases. So initially we just did a survey study where we looked quickly at, we tried to identify from a large suite of chemicals. A lot of these have been talked about earlier today in a number of different studies. Using, we have things like caffeine, but also anticonvulsant drugs like carbamazepine, hormones like estradiol, antibiotics, lincomycin, ciprofloxacin, sulfamethoxazole, and what we found is that after doing the study was those that are highlighted here we found exhibited moderate or high reactivity with permanganate. It might be under the time scales we would use in a treatment plant. Maybe we should be looking at whether or not they are degraded in these situations. But as you can see, permanganate is a selective oxidizing agent. It's not something that's going to be used to just take out everything. So if your goal is to remove everything from the water, you may probably want to look at something that's more like an advanced oxidation technology. However, if your plant is using permanganate, you might get some benefit from that for things like carbamazepine. And also, if we're thinking about the overall fate of these chemicals in the treatment plant, we would want to, during subsequent processes, we might want to be looking at how the byproducts of permanganate reactions are influenced. So after doing this, we undertook much more extensive kinetic studies under a wide range of conditions to try to figure out develop a model that might be predictive in treatment plants. And so just to show an example, this is from some results with carbamazepine, which is the anticonvulsant drug, which is widely detected in surface waters. And so what we did in really nice controlled laboratory studies at relatively high concentrations, we did a number of kinetic studies and established a kinetic model. And our goal was, we said, well, how well will this model work in predicting treatment under more realistic conditions with, I guess the most important thing is realistic doses of permanganate. In the lab, we work in hundreds of micromolar, which corresponds to tens or hundreds of milligrams per liter of permanganate. But a real treatment dose is usually 0.5 to 4 milligrams per liter permanganate. And so what we did, though, is we were able to get some utility waters from some Illinois utilities, and we took our model and we predicted a certain profile for this treatment conditions here. And although this is our initial concentration we spiked into these as carbamazepine was 10 micrograms per liter, that's higher than what you would find in surface water. It's still very, very, very much lower than what we were using in our lab studies, which was about maybe 500-fold higher than this in those lab studies. And kinetics are kinetics, and if our rate laws extrapolate over 3 or 4 orders of magnitude, it's reasonable to think they might extrapolate over 1 or 2 more orders of magnitude. But anyways, what we found is we found very good agreement between our model developed from nice, clean laboratory solutions and what we can observe in real surface source waters. And so that was nice. And we've also done a number of studies with tandem mass spectrometry and NMR techniques to try to identify byproducts from these reactions and then propose reaction mechanisms. But I think for today's discussion, I won't go into those. But I'll be happy to talk about it. So that's a conventional process, but we're also interested in thinking about maybe technologies to the future. And one of the things that we're interested in our center is trying to get away from chemically intensive processes and try to use treatment approaches that would not rely on chemical inputs like chlorine in our water. And one of the things that I looked at in collaboration with some other researchers in the center, was using photocatalytic technologies where we use semiconductor materials like titanium dioxide, which when illuminated with ultraviolet light generate reactive oxygen species at their surface that then can oxidize contaminants. And so what we saw initially was not unexpected from what we know about photocatalysis, is that under UV irradiation, these materials, we saw that these materials could effectively transform. So this is just the concentration of Cipro, a widely used fluoroquinolone, as a function of time. And we see that it's rapidly transformed under ultraviolet. And that's not surprising from what we know about that. But what was a little more surprising from our study was that we saw that even when we used wavelengths that were more into the visible, only allowed visible light to hit the materials, and we know titanium dioxide materials are not excited by visible light, we still saw transformation of the fluoroquinolones. And so that was a little bit sort of atypical behavior. But what's really, from my standpoint, when I think about selective treatment, this is nice because not many chemicals are susceptible to such visible light-mediated processes. So it's possible that if fluoroquinolones are really what we're concerned about, and maybe in some waste, maybe an industrial waste of fluoroquinolones rich, we might be able to use this to treat those really low levels of fluoroquinolones in the presence of much more concentrated background levels of other constituents. And we did a number of studies where we looked at using different techniques, and we identified the mechanism of this was, and I'm not going to go into this too much detail, but essentially we had a surface complex mediated we were photoexciting the antibiotic on the surface of the semiconductor material. What happens when it adsorbed to the surface, its spectrum shifted into the visible light so it could absorb photons in the visible range. And as a result of that, it underwent oxidation, a mild oxidation, just small changes in the structure of the molecule. But as you'll see later, that did result in a loss in antimicrobial potency of the molecule. So the third technique I want to talk about is just another technology we've been looking at, which is using surface-supported or supported metal catalysts in combination with hydrogen, which is to generate potent reducing agents. So instead of oxidizing, which the other process I've talked about have been trying to oxidize the contaminants, there are a number of micropollutants that are not really readily amenable to oxidation. But what makes them resistant to oxidation, we thought, might make them susceptible to reductive transformation to break them down. And so we found, we've been using sort of supported palladium and nickel catalysts. And we've been interested in our work to date on kinetics and mechanisms, but we're also starting to look at how these sort of materials-based technologies can stand up to fouling and issues like that as we go into the future. What's nice about reductive processes is compared to oxidations, they're much more compound selective. They're not so broad. And we focused our studies on two classes, N-nitrosamines, like NDMA, if you've heard about. This is a disinfection byproduct of chloramination processes, particularly in wastewater treatment plants that have a lot of organonitrogen precursors in the water. And also iodinated X-ray contrast agents. And those of you that are interested in trying to assess the inputs of wastewater into natural surface water bodies, you may want to look at X-ray contrast agents. These triiodinated compounds are very resistant to any kind of conventional wastewater treatment processes and activated sludge, and they also don't absorb to things, so they fly right through the plant. They go into the surface waters, and they're pretty much stable in surface waters. Caffeine has been used as a tracer in a lot of places, but I think caffeine is a little more biodegradable. These compounds can be used as kind of a really good tracer in surface waters for the contributions of wastewater effluent. So, but because they're very resistant, we thought, though, looking at their halogenated structure, they might be amenable to dehalogenation. And in fact, that's what we found. We found in our study, we found complete dehalogenation pretty rapidly. And once we removed the halogens, I haven't done this, and I wouldn't mind I would like to work with somebody on this, is I would anticipate that the final product is much more biodegradable than is the parent material, which is almost unbiodegradable by aerobic sort of heterotrophs. So we found that, and I'm just going to show one plot from this. We did some work where we were looking at just some simple batch studies looking at diatrozoate as a function of time in a wastewater treatment effluent, secondary. Well, actually, I guess it had gone through nitrification, our local wastewater treatment plant here. And what we found is that compared to deionized water, our rate of transformation was much lower in the wastewater effluent. However, in that wastewater effluent had something like 5 milligrams per liter dissolved organic carbon. But if we had maybe done some pre-treatment with granular activated carbon in that water, we can lower the DOC levels to a point where essentially the GAC treated water was again back to our catalytic process was just as effective. And now I don't believe, though ideally we wouldn't want to have to remove all the DOC in the wastewater effluent, but it may be that there's only like a small fraction of the DOC that's really causing the problems in this process. And if we can identify what those are, maybe we can design something that's more selective just to pre-treat those things before we want to remove these. And then one final slide, just as we go on, any process we want to look at, we want to look at, again, the energy efficiency of those processes, and also are we doing anything? If we're transforming the chemical, are we actually having a benefit here? And so what I'm plotting here is just as a function of the amount of irradiation energy, these are for photochemical and photocatalytic processes. So this axis is just the amount of energy input from lights into the process. And on this axis, we have two things plotted. One is just the concentration, and so we can see for UV photocatalysis, visible light photocatalysis, and UV just photolysis with no material present, how much the dark symbols are the concentration of the parent compound remaining for different doses applied of light. But the other thing we're plotting are these PEQs, and what this is just essentially it's the norm, it's the overall potency, antibiotic potency of the product mixture, including the remaining parent compound and any byproducts formed. And what we see is that potency, which is measured by just microbial cell growth inhibition assays, what we see is that essentially for at least for these different photochemical processes, that when we get when you degrade 50% of the parent compound, you also lose 50% of the potency. And this means that probably any byproducts that we're forming, and we're not probably in most of these we're not mineralizing the chemicals. Those byproducts, though, are probably at least tenfold less have tenfold less potency. And so I think we need to be, as we're going in the future, couple these kinds of studies, although these are just simple in vitro assays. There's a number of other tests that have been talked about today, I think, that could be applied. So just my thoughts on challenges to treatment of waters containing micropollutants. The first is this question of removing the drop. Somebody else told me this: removing the drop of poison in the ocean of water. We're trying to treat nanogram per liter levels of these pharmaceutically active compounds in water that contains 6 orders of magnitude, or maybe 4 or 5 or 6 orders of magnitude higher amounts of other organics in those waters. And so how do we treat those chemicals without having to just put in so much energy to take everything out? I mean, we can do that. We can just make ultra-high purity water, right? But that's going to take a lot of energy and be very costly. The other question is, how low should we go? 10 years from now, I'll probably be sitting in this room and people will be talking about PPQs. And then the question is, okay, just because we can detect parts per quadrillion in everything that we have, is that a problem? Where we need to get a sense of where we're going to establish limits here, because analytically we're moving ahead pretty fast, and we're always going to beat out. We're always going to stay ahead of any plans. We've talked about a lot of different pharmaceuticals. Are we going to go after all of them, or maybe we need to think about which ones of these or which of their metabolites we really want to go after? Again, maybe hormonal compounds, which have been documented to have effects at nanogram per liter levels to aquatic life in near outflows of wastewater treatment plants. Maybe we should be thinking about those, but are we really worried about ibuprofen at nanogram per liter levels? And then any solutions that we come up with should be, maybe this buzz term of the day, sustainable. That means they should be energy efficient. We should not be devoting high levels of energy to maybe things where we don't know if there's really an added benefit. And then we want to think about maybe limited chemical inputs. So this might include physical separation processes like reverse osmosis and other membrane processes, and also things of the nature like I talked about today, heterogeneous catalytic processes that maybe are, especially if we can use something like sunlight to drive photocatalytic processes, this could be promising. But certainly there's a lot of hurdles to overcome before these would be ready for adoption as technologies. And the people who did the real work, my students, they're working on both reductive catalytic processes and also oxidation strategies, and be happy to try to answer any questions.